Welcome to the Longevity Papers podcast.
It is so good to be back.
It really is. It feels like it's been a minute, doesn't it? We have been officially on hiatus since, mid-December 2025.
That's right. And looking at the calendar now, it's February 10th, 2026. We completely skipped January.
We did, but I think for a very, very good reason. We weren't just on vacation.
Not at all. We spent the last, 6 weeks doing what we always promised to do for you. Digging through the noise to find the signal. And I have to say, the stack of papers we reviewed this time was massive.
I mean, what was the final count? I lost track.
Over 140 distinct manuscripts that were published between mid-December and, now.
140. That's a lot of reading. And I think we need to be really upfront with everyone listening. How many of those actually made the cut for today's discussion?
5.
Just 5.
Just 5.
Wow. That is a brutal attrition rate.
It is, but it has to be. When we sat down to plan our return for the new year, we sort of recommitted to our core philosophy. We really did.
Yeah. We had a real heart-to-heart about it.
We're just not here to talk about a study that shows, a 0.1% improvement in a worm's lifespan using a method that will never, ever translate it to humans. That's not the game we're playing.
No. We're hunting for 3 specific things. And we really held ourselves to this. We're looking for credible institutions, first and foremost.
Yep. Then, novel mechanisms. And by novel, we mean something that genuinely makes you rethink the biology, not just another paper on a pathway we've known about for 20 years.
And crucially, the third thing is human translatable data, or at least a clear path to it.
Exactly. We're deeply, deeply skeptical of incremental gains. We're looking for those order of magnitude shifts, the papers that make you stop, put down your coffee, and say, okay, we actually need to change our model of how this system works.
And the vast majority of that stack of 140 papers, if we're being honest.
It was noise. It really was. Marginal contributions, duplicate data, things that were frankly just not that interesting or didn't move the needle. But these 5...
These 5 are the signal?
They are. Each one of them forces a model change in how we view aging biology.
So that's the mission for today. We're going to unpack 5 specific papers published between December 2025 and mid-January 2026. And we've got quite a lineup. We're talking everything from spider venom to garlic and even a functional cure for HIV that might just rewrite the rules of immune aging.
It's a fascinating stack. I'm excited. Let's get into it.
All right, let's do it. Let's unpack this first one. We are starting with a paper that came out of Massachusetts General Hospital, a real powerhouse. It was published in bioRxiv, and just a note for everyone, that's pronounced bioRxiv, on December 16th. And the title is Selective Activation of NAVI 1.3, Restores Lymphatic Contractility in Age and Injury.
This is a perfect example of what we were just talking about. It's looking when no one else is really looking and finding something huge.
Right. So the context here is the lymphatic system. And I feel like for the longest time, the lymphatics were treated like the poor cousin of the circulatory system.
Yeah. Oh, absolutely. The forgotten plumbing. Everyone cares about the heart, the arteries, the veins, the blood vessels. But the lymphatics are just there, ignored.
But that's changing, isn't it?
It is. In the last few years, lymphatic dysfunction has been formalized as an actual legitimate hallmark of aging. And you have to think of the lymphatics as the body's sewer and recycling system. It's not just passive drainage.
No, it relies on intrinsic pumping, right?
Yes.
The vessels themselves are contracting.
Precisely. They have these little muscle sections that create pulsations, and that's what moves the fluid along. That's what clears out all the metabolic waste, the dead immune cells, the excess interstitial fluid. It's an active process.
And like any mechanical pump, I guess, it just wears out over time.
It does. The research is pretty clear on this. That pumping capacity, what they call contractility, it declines by about 50% as we age.
50%.
50. So imagine if your city's entire waste management system suddenly started working at half capacity.
It'd be a disaster. You'd get backup, trash piling up in the streets.
Exactly. In the body, that translates to fluid buildup, think in edema, but also poor immune trafficking. The immune cells can't get where they need to go. And you get this accumulation of metabolic toxins around your cells. It creates a dirty cellular environment.
And the problem, as I understand it from this paper's introduction, is that we haven't really had a good way to fix it. We didn't have a drug that could just, turn the pump back on.
We didn't. And the reason for that is specificity. It's a huge problem. If you try to stimulate muscle contraction broadly with a drug, you're going to hit the heart. You're going to hit the smooth muscle around your arteries.
And that's bad.
That's very bad. You can't just ramp up contractility everywhere without causing a heart attack or massive hypertension. So For a long time, this was considered an undruggable decline, which
Ah.
just didn't have a target that was unique to the lymphatics.
Until now. And this is where Ruscic and the team at Mass General found something really, really surprising. They found Navi 1.3.
Yeah, Navi 1.3. It's a voltage-gated sodium channel. Now, normally if you ask a neurobiologist or a developmental biologist about Navi 1.3, what are they going to tell you?
It's a fetal thing or something you see after nerve injury. It's not supposed to be in healthy adult muscle tissue.
Exactly. It's supposed to be turned off, but they found it.
And not just anywhere.
No. This is the aha moment. This is the part that makes this a model-changing paper. They found it expressed selectively in adult lymphatic muscle. But here's the kicker. It was absent in the heart. It was absent in vascular smooth muscle. And it was absent in the mature brain.
It's the holy grail of drug development, isn't it? Finding a target that is only on the thing you want to fix and literally nowhere else important.
It is. It offers a clean drug target. It's
Yeah.
a handle you can pull that, theoretically, only turns on the lymphatic pumps, leaving the heart and arteries completely alone.
So they found the lock, and then they needed a key. And this is where the paper gets really interesting and honestly a little bit scary. They used a compound called TF2.
Yes, TF2, which is a peptide derived from spider venom.
Of course it is, because nothing says rejuvenation quite like spider venom.
Well, nature is often the best chemist, right? And spider venom is just packed with these incredibly potent peptides that mess with ion channels. That's how they paralyze their prey. But in this case, TF2 is a very specific activator of Navi 1.3.
Okay, so they had the target, they had the activator. What happened when they actually gave this spider peptide to aged mice?
The results were, and I'm not exaggerating, striking. In the aged mice, and remember, these are the ones with that 50% decline in pumping, this single peptide fully restored lymphatic pumping back to youthful levels.
Wait, not partially restored, fully restored.
Fully. They measured what they call the lymphangin ejection fraction. It's just a fancy way of saying how much fluid the vessel pumps with each beat. It went right back up to where the young mice were, and as a result, it massively accelerated the clearance of interstitial fluid.
So they basically unclogged the entire sewer system and got it running at full capacity again.
With a single dose, yes.
That is incredible.
Yeah.
And it wasn't just in the context of aging, right? I saw they also looked at injury models.
They did. This is really relevant clinically. They looked at radiation injury, which is known to just decimate lymphatic pumping. It's a huge problem for cancer patients undergoing radiotherapy. And TF2 partially rescued that deficit as well.
So what does this all mean, big picture? If we can reliably turn the lymphatics back on, are we just talking about reducing, swollen ankles in the elderly, or is this bigger?
It's so much bigger. I mean, think about it. If you fix the drainage system, you are potentially fixing the local environment for every single organ in the body. We're talking about clearing neuroinflammation from the brain because the brain has its own lymphatic connection, the glymphatic system.
Right.
We're talking about clearing metabolic waste accumulation from muscles, from the liver. If this holds up, it opens up a completely new therapeutic node for aging. It's not just about fluid, it's about the fundamental cleanliness of the cellular environment.
Okay, and there's always a but, we have to talk about the skepticism. This is a spider venom peptide. I can't imagine we're going to be popping spider pills anytime soon.
No, and that is the major immediate critique here. TF2 is a peptide that presents delivery challenges. You can't eat it. Your stomach acid would just digest it into amino acids.
So it has to be injected.
It would almost certainly need to be injected. And beyond that, even though they showed Navi 1.3 isn't in the adult heart or brain, we have to be extremely careful with off-target effects. Spider venom derivatives are incredibly potent. You need rigorous screening to ensure it isn't hitting some other unknown ion channel we haven't thought of.
Yeah, and even if they say it's absent in the brain, biology is always messy. Absent could mean very, very low levels that might still be a problem.
Correct. But what's important is that the concept is now validated. Even if TF2 itself never becomes a drug, they've proven that Navi 1.3 is the handle. Now, medicinal chemists can go to work designing small molecules that target that specific handle, and those might be orally bioavailable and even safer.
So it provides a new roadmap.
Exactly. It connects to this broader field of vascular rejuvenation, therapies like VEGF. But this is totally distinct because of that beautiful ion channel selectivity. It's like using a surgical strike versus a carpet bomb.
A surgical strike on the body's sewer system. I love it. Okay, let's move from the exotic world of spider venom to something you can probably find in your kitchen right now. Let's talk about the garlic paper.
Ah, yes. This one is fascinating because it takes something we all sort of know from folk wisdom, and apply some really rigorous modern biochemistry to it.
This paper comes from CABIMER, which is the Andalusian Molecular Biology and Regenerative Medicine Center in Spain. It was published in Cell Metabolism on December 15th. The title: Enhanced Non-Enzymatic H2S Generation via Dialyse Sulfurated Compounds Extends Lifespan and Health Span in Mice.
And the most important word in that whole title is non-enzymatic. That's the whole story.
We will definitely get to that, but first, let's set the stage with something called gasotransmitters.
When I think of gas, I think of, well, never mind that. But biologically, we're talking about signaling molecules, right?
Yes, exactly. Most people have probably heard of nitric oxide, or NO. It's a gas that tells blood vessels to relax, which lowers blood pressure. But there's another major one, hydrogen sulfide, H2S.
Which unfortunately smells like rotten eggs.
It does, but in tiny concentrations in the body, it's a critical signaling molecule. It's a master regulator of mitochondrial bioenergetics, so how your cells make energy, and it's a huge player in our native antioxidant defense systems. The problem is, like almost everything good, H2S levels drop pretty significantly as we age.
Okay, so we have less of this protective gas. So previous attempts to fix this have tried to just, what, pump more H2S in or boost the enzymes that make it? Exactly. The body has a set of enzymes that naturally produce H2S from amino acids like cysteine. But as we get older, those enzymes start to fail or get downregulated. So if you try to boost the enzyme, you're essentially whipping a tired horse. It just doesn't work that well.
And this is where the garlic paper comes in with a totally different approach. They use these diallysulfurated compounds,
Which are basically these sulfur compounds naturally found in garlic, onions, and other allium vegetables. But the real brilliance of this study was the mechanism. These compounds don't need the body's failing enzymes to work.
They bypass them.
They bypass them completely. They undergo a direct chemical reaction in the body with other biological molecules like glutathione to release H2S. It's a non-enzymatic source. It doesn't rely on the failing machinery at all.
So it's like bringing your own portable generator to a house during a blackout, instead of trying to fix the main power plant.
That is a perfect analogy. You're providing an independent source of a vital gasotransmitter.
So they gave these garlic-derived compounds to mice. What happened?
The top-line result is a lifespan extension in wild-type male mice of approximately 10 to 15%.
10 to 15%. I mean, that's solid. It's not immortality, but that's a statistically significant bump.
It's a very solid hit for a single intervention in a mammal, but I'd argue the health span data was even more interesting. They saw improved glucoregulation, so better control of blood sugar. They saw improved locomotor functions, so the mice were physically more capable and active. And they saw improvements in neurocognition.
And they were able to nail down a specific biomarker for this, right? Something they called persulfidation. What is that?
Yes, this is the core mechanism. H2S causes this post-translational modification on proteins called cysteine persulfidation. The simplest way to think about it is like a chemical shield. The H2S molecule adds an extra sulfur atom to cysteine residues on proteins.
And that protects them.
It protects them from irreversible oxidation damage. It effectively armor plates the proteins so they can keep functioning properly in a high-stress, aged environment. It's a protective shield.
And they didn't just stay in mice for this one, which is great to see. They actually looked at some human data, too.
They did. It was a correlation study, so we have to be careful, but it was very suggestive. They looked at a cohort of older, sicker humans with multiple pathologies, and they found that the individuals who naturally had higher levels of this protein persulfidation,
They were doing better.
they were doing better. They had preserved muscle strength and lower triglycerides. It's a nice piece of evidence that connects the molecular mechanism in mice to a relevant clinical outcome in humans.
So the mechanism seems to hold up across species. High persulfidation seems to equal better function.
It certainly points in that direction, yes.
Okay, let's get to the expert analysis. Is this paper just a very, very complicated way of telling us all to eat more garlic?
It's tempting to say that, and it's probably not bad advice, but there's more nuance here. The paper argues for specific sulfur compounds at specific bioavailable levels. To get the equivalent dose they used in mice, you might have to eat an absolutely enormous amount of raw garlic every day.
Probably not great for your social life.
Not at all. But what it does is it validates the epidemiological data that diets rich in these allium vegetables are generally protective. Now we have a specific molecular reason why.
And the skepticism. You mentioned earlier the study was only in male mice.
Yes, and this is a recurring and frustrating issue in longevity research. The entire lifespan study was done in male mice only. We absolutely need to see the female lifespan curves. We know that females metabolize sulfur compounds differently, and their baseline antioxidant defenses are not the same as males. It's a huge gap in the data.
And it's still a single intervention.
Right. A 10 to 15% lifespan extension is great, but we're always looking for things that can be stacked. The big question for me is, what happens if you combine this H2S donor with an NAD plus booster or with rapamycin? Do you get an additive effect? 20%? 30%? Or do they interfere with each other? That's the next experiment I really want to see.
Still, the core idea of a non-enzymatic way to restore a youthful signaling molecule, that's a model change. It means we don't always have to fix the broken machine. Sometimes we could just build a bypass around it.
It really validates the whole gasotransmitter theory of aging. It's not just about genes and proteins. It's about these small, transient signaling molecules that orchestrate everything.
All right, moving on to paper number 3. We are going from protective gas to so-called zombie cells. This one comes from the Mayo Clinic, published in Nature Cell Biology on December 15th. The title is Mitochondrial RNA Cytosolic Leakage Drives the SASP.
This is a heavy hitter. The Mayo Clinic is, it's ground zero for senescence research. When they publish something in this area, you pay attention.
Okay, before we dive in, let's do a quick refresher for everyone on cellular senescence and the SSSP.
Right. So senescent cells are cells that have suffered some kind of damage, usually DNA damage, and have stopped dividing. But crucially, they refuse to die. They just hang around in the tissue.
Like zombie?
Exactly. And they're not just inert. They actively secrete a toxic cocktail of inflammatory chemicals. We called this the SASP, the senescence-associated secretory phenotype. This SASP leaks out, irritates neighboring cells, causes chronic inflammation, and basically degrades the function of the entire tissue.
It's like having one really angry, screaming neighbor on your street that makes life miserable for everyone else.
A perfect analogy. And for a long time, we thought we knew why they were screaming. We thought the alarm was being pulled by mitochondrial DNA.
Explain that old model.
The old model, and I want to be clear, it's still valid, it's just incomplete, was that the mitochondria in these old, damaged cells get leaky. They start to fall apart. And they spill their contents, including mitochondrial DNA, or mtDNA, into the main part of the cell, the cytosol.
And the cell freaks out.
The cell freaks out. It sees this DNA where it shouldn't be, and it has an ancient alarm system that thinks it's a virus or a bacterium invading. That alarm system is called the CGAS-STING pathway, and when it gets triggered, it's one of the main drivers of that inflammatory SSP.
Okay, so that was a textbook explanation. But this paper from the Mayo Clinic says, hold on, it's not just the DNA.
Exactly. Victorelli and the team found that it's not just mtDNA leaking out. Mitochondrial RNA, mtRNA, is also leaking out of these damaged mitochondria.
And that pulls a completely different alarm handle.
A completely different but parallel alarm system. This leaked mtRNA triggers sensors called RII and MDA5. And these are sensors that the body's immune system usually uses to detect RNA viruses, like the flu or, coronaviruses.
So the senescent cell is effectively tricking its own internal systems into thinking it has a viral infection.
That's precisely what's happening. The leaked mitochondrial RNA looks structurally similar to viral RNA, so these sensors grab onto it. This activates a whole other inflammatory cascade, leading to something called MAVS aggregation, which is another powerful engine for pumping out the SAP.
Okay, so why does this distinction between DNA and RNA matter so much? Why do we care if the alarm is CGA-STING or RICI and MDA5?
It matters enormously for developing therapies. For one thing, it provides a dual pathway model for this sterile inflammation. It explains why some drugs that only block the DNA pathway might not have worked very well. You were only cutting one wire to the alarm. The other one was still blaring.
It identifies new drug targets.
Exactly.
Yeah.
This brings us to the whole concept of xenomorphics.
Right. So senolytics are drugs that kill the zombie cell. Xenomorphics just try to tape its mouth shut.
A great way to put it. Xenomorphics try to stop the SASP without killing the cell. And this paper suggests that if we develop inhibitors for IZI or MDA5, we could potentially quiet down that inflammatory screaming without having to get rid of the cell itself.
Which could be a lot safer, couldn't it? Because killing cells, senolysis, that can leave holes in the tissue, right?
Senolysis definitely has risks. You can get off-target cell death, you can have problems with tissue repair, or you might be destroying cells that, even though they're senescent, are still providing some important structural integrity. If you can just quiet them down, that might be a much more elegant and safer strategy for maintaining tissue function as we age.
The paper also identified the actual pores that are letting this RNA out proteins called BX and BA.
Yes, and this is another fascinating twist. BX and BA are famous proteins, but they're usually associated with apoptosis, which is programmed cell death. They're the executioners. They punch huge holes in the mitochondria to kill the cell.
But here, they're not killing it.
No, it seems that in senescence, they're just punching smaller transient holes, just big enough to let some RNA out, but not big enough to trigger cell death. It's what they call a sublethal release.
That's just wild. The cell is using the literal machinery of death, but not to die. It's just using it to scream for help.
It raises a really deep philosophical question about senescence. Is the SASP purely a destructive process? Or is it actually a cry for help that just gets stuck in a chronic loop in old age?
And what's the expert skepticism on this one? Any caveats?
The main one is the model system. The study focused very heavily on the liver, specifically in MSH models, which is metabolic dysfunction-associated steatohepatitis. It's a disease model. We need to see this proven in other tissues that are critical for aging, like the brain and the heart.
Does this RNA leakage happen in senescent neurons, in senescent cardiomyocytes?
Exactly. And does blocking it in those tissues actually extend health span or lifespan, or does it just temporarily reduce inflammation? Those are the next big questions.
Still, knowing there's a second parallel alarm system is a huge model change. It explains why some previous attempts might have failed. They were only cutting one wire of the alarm when there were two.
Precisely. It complicates the picture, but in a way that gets us much closer to the biological truth.
Okay, let's move to paper number 4. This is the wild card of the bunch. And honestly, this is the one that completely blew my mind when I read it.
I had the same reaction.
It's from University College London and a company called Scent Cell, published as a preprint on bioRxiv on January 8, 2026. The title is Awakening Intracellular Immunity for Functional HIV Cure via CD4 T-Cell Reprogramming.
And I can hear the listeners right now asking, wait a minute, isn't this a longevity podcast? Why on earth are we talking about HIV?
That was my exact first question, too. But the connection to aging is profound and undeniable.
It is on 2 levels. First, on a direct practical level, HIV acts as a massive accelerant for aging. It's a model of premature immunosenescence.
Explain that.
Well, patients with chronic HIV infection, even if it's well controlled with drugs, often have an epigenetic age that is about 13 years older than their chronological age. The virus causes so much chronic immune activation and inflammation that it wears the system out.
So they get old people diseases much earlier.
Exactly. They get heart disease, frailty, cognitive decline, certain cancers in their 40s and 50s. So finding a functional cure for HIV is, in a very real sense, a longevity intervention for those 40 million people living with it. It's about restoring 13 years of lost immune lifespan.
Okay, that's the first connection. What's the second, bigger one?
The second is that the mechanism they discovered has breathtaking implications for every single one of us, HIV positive or not. It's a paradigm shift.
Let's get into that mechanism. The title uses this phrase, awakening intracellular immunity. What does that mean?
Okay, so the standard problem with curing HIV is that the virus hides. It integrates its DNA right into the genome of our own CD4 T cells, and then it goes dormant, creating a latent reservoir. The immune system can't see it, and our drugs can't reach it when it's not active.
Right.
So what this team at UCL did was something truly radical. They took these T cells from patients, and in a dish, they transiently reprogrammed them using techniques related to Yamanaka factors.
And this reprogramming did something unexpected.
It did something that sounds like pure science fiction. This reprogramming activated what they call an antiviral telomere transfer program.
Hold on, you have to unpack that phrase, telomere transfer program.
I know, it sounds crazy. What they describe is that the CD4 cells, upon being reprogrammed, initiated a dormant, built-in program where they use telomeric material, the protective caps at the end of our chromosomes, or at least telomere-associated machinery, to physically find and excise, to cut out the integrated viral DNA from their own genome.
Let me get this straight. The cell has a dormant program that allows it to use its own telomere system to find and delete foreign DNA that has been spliced into its own chromosomes.
That is the claim. And the results they present are just staggering. Ex vivo, in the Petri dish, they saw clearance of these HIV DNA reservoirs within hours to days.
Hours to days, not weeks or months.
Hours to days. And this is without any antiretroviral drugs. Then they took these reprogrammed cells and put them into humanized mice that were infected with HIV. The reprogrammed T cells went in, eliminated the virus in the animal, and maintained undetectable levels for months.
That is, by definition, a functional cure.
It's the closest thing to a scalable functional cure that we have ever seen. I mean, previous attempts like shock and kill or even CRISPR-based approaches have had modest efficacy at best or huge safety risks. This paper is claiming greater than 90% elimination by awakening the cell's own dormant machinery.
Okay, this brings us to the massive longevity angle for everyone. If our own immune cells have a dormant program that can recognize and delete foreign DNA from our genome,
You see it. What else can it delete?
could it delete other viruses? What about CMV or EBV?
Exactly. Cytomegalovirus, or CMV, and Epstein-Barr virus, EBV, are two other latent viruses that are massive drivers of what we call inflammaging. Almost everyone has them by the time they're old. They simmer away in the background, constantly poking the immune system and causing it to become exhausted.
So if this telomere transfer mechanism can be harnessed to clear those latent reservoirs,
You could potentially rejuvenate the immune system of the entire elderly population. You could remove a huge source of chronic inflammatory burden that we all carry. It's a staggering thought.
awakening a sleeping giant within our own cells to literally clean house.
It is. Now, we have to inject a heavy dose of caution here. This is a preprint. And elite-like control is a very bold claim. We absolutely need human safety trials, which, to their credit, the paper mentions are imminent, with a phase 1 planned.
The big risk has to be off-target cutting, right?
Yeah. That's the billion-dollar question. You need to make sure this cutting mechanism is exquisitely specific to viral DNA and doesn't accidentally start cutting out important parts of our own human genome. You don't want the editor to get too aggressive and start deleting chapters of the instruction manual.
Right.
Genomic stability is paramount, but the shoe novelty here is off the charts. It shifts the entire paradigm from let's keep the virus quiet with a lifetime of drugs to let's reprogram the cell to delete the invader for good.
It really feels like we're moving from chemical engineering, designing drugs, to software engineering. We're learning how to rewrite the code of the cell itself.
That's where the future of medicine is heading, without a doubt.
All right, wow. We have covered spider venom, garlic, zombie cell alarms, and cellular telomere editors. Let's bring it home with the fifth and final paper. We're calling this one the anchor. And it's special because of the species that was studied.
Humans. Finally. Real, live, aging humans.
This paper is from the University of Oxford, published in the top-tier journal Nature Aging on January 16th. The title: Rapamycin exerts its geroprotective effects in the aging human immune system by enhancing resilience against DNA damage.
This is, without a doubt, the paper the entire longevity community has been waiting for.
We talk about rapamycin constantly on this show. It's the most robust chemical geroprotector we have. It works in yeast, worms, flies, mice, even dogs. But we've been lacking that definitive mechanistic study in healthy aging humans.
We've had observational data or studies on kidney transplant patients who are taking these huge immunosuppressive doses, but this was different. This was a proper placebo-controlled trial in older adults looking specifically for geroprotective effects at a low dose.
And the dosing is absolutely critical here, isn't it?
It's everything. They used a low dose, a non-immunosuppressive regimen. This is the anti-aging dosing schedule that people talk about, usually taken just once a week, rather than the high daily dose used to prevent organ rejection.
So what did they find? What is rapamycin actually doing in older people?
Well, the classic textbook assumption has always been that it's all about autophagy. We thought rapamycin inhibits a pathway called mTOR, which then triggers autophagy, the process where the cell cleans up its own internal trash. And while that certainly does happen, this paper found a different, and perhaps more important, primary mechanism in humans.
It enhanced the cell's resilience against DNA damage.
Directly. It didn't just help clean up the mess after the fact. It seemed to stop the mess from causing a downstream panic. Specifically, they looked at a biomarker called P21.
And P21 is a classic marker of senescence, right? It goes up when a cell gets stressed.
It's a key marker of DNA damage-induced senescence. When a cell suffers significant DNA damage, P21 levels shoot up, and that's the signal that tells the cell to shut down division and become senescent. In the immune cells of the patients treated with low-dose rapamycin, P21 was significantly reduced compared to placebo.
So the cells were still experiencing the stresses of being in an old body, but they weren't flipping the zombie switch as easily.
That's the perfect way to describe it. In fact, they did a follow-up experiment where they took T cells out of these patients and deliberately exposed them to stress in a dish. The cells from the rapamycin-treated group had what they called a reduced lesional burden. They either accumulated less physical damage to their DNA in the first place, or they were repairing it much more efficiently.
That is such a subtle but powerful shift in thinking. It's not just about cleaning up the garbage. It's about making the cell tougher, more resilient.
It's all about resilience. And for you, the listener, this is by far the most immediately actionable paper in the whole stack today. Rapamycin is an FDA-approved drug. It's off patent. It costs about $2 a month.
But, and this is a big but, you can't just go buy it at the corner store. It requires a prescription and medical supervision.
Absolutely. No one should be doing this on their own. But what this paper does is it provides the rigorous biological proof of concept that cautious, evidence-based doctors have been waiting for. It confirms that this low-dose weekly schedule acts as a xenomorphic, calming down those senescent signals, rather than as a dangerous immunosuppressant.
So it validates the potential safety and the mechanism for this specific use case.
It does. And it ties directly into the big upcoming clinical trials like the TAME trial for metformin and the larger rapamycin longevity trials that are now getting underway. This is the human data that provides the justification for those bigger, more expensive studies. It's the bridge from animal models to legitimate clinical practice.
So to sum it up, we now have a confirmed human mechanism-enhanced DNA damage resilience. We have a clear biomarker-reduced P21. And we have a drug that is available right now.
It moves the conversation about rapamycin out of the realm of biohacker speculation and firmly into the world of evidence-based clinical investigation. It's a huge step forward.
Okay, that is the stack. 5 papers. Let's take a moment and just zoom out and synthesize this. We started with a pile of over 140 papers, most of which we dismissed as noise. We ended up with these 5 signals. What's the through line here? What's the meta-narrative?
I think the overarching theme is a clear shift in the entire field, from a focus on simple lifespan extension to a much more sophisticated model of systemic repair. We aren't just looking for a single gene that makes a worm live 20% longer anymore. That's old news.
Right. Looking at these 5, we're talking about repairing the body's sewer system with the lymphatics paper. We're talking about bypassing broken enzymes to restore a signaling gas with the H2S paper.
We're discovering brand new alarm systems for inflammation with the mitochondrial RNA paper. We're exploring how to rewrite a cell's own software to fight invaders with the HIV and telomere paper.
And we're finally validating one of our best tools, rapamycin, as a resilience agent directly in humans.
The level of sophistication has just ratcheted up enormously. We're moving from finding a gene to reprogramming a system. The HIV paper really is the poster child for this. It's not about a new drug. It's about unlocking a capability the cell already possesses but has forgotten how to use.
And I think it's important to say that while none of these papers promise that you or I are going to live to 150 starting tomorrow.
Oh, absolutely not. No single paper here promises a 10-plus year life extension for a healthy human yet. But the mechanisms we're uncovering, Navi 1.3, targeting, non-enzymatic H2S donation, mtRNA sensing, telomere transfer, these are the fundamental building blocks for that eventual leap. You can literally see the toolkit for radical rejuvenation coming together.
So here's my final provocative thought for you, and for everyone listening. I just can't stop thinking about that HIV paper. If our immune cells have a dormant, built-in telomere transfer program that is capable of deleting foreign DNA, what else is that program capable of?
That is the ultimate question, isn't it? If we can learn to control that awaken signal, could we direct that intracellular immunity to clear not just viruses, but our own accumulated genomic damage? Could we tell it to delete the stuttering genetic repetitions that cause Huntington's disease?
Could we teach it to recognize and excise the specific oncogenic mutations that drive cancer before a tumor even has a chance to form?
If the software is already there, dormant inside our cells, maybe we just need to learn the password.
I think we're just starting to type it in.
I think you're right.
And on that note, we are officially back. We will be sifting through the stack again next week, so you don't have to.
Always looking for the signal in the noise.
See you next week for more papers.